All right, so today we're going to dive into something pretty deep, even for us. We're not just looking at how scientists, figure out the evolutionary history of life. We're really getting into some serious philosophical territory with this one.
Yeah, you hit the nail on the head there. We always love to take a scientific field and see what kind of weird philosophical questions come up
That's
for it.
exactly.
And phylogenetic inference, I mean, trying to figure out those evolutionary relationships, it just brings up so
What's that
many.
then?
It makes you think about how we even understand the past at all, what makes something good evidence, even what those things we're studying actually are.
Yeah, and I think for our listeners, the people who really want to get this but don't want to wade through a whole textbook, this is going to be a really focused exploration.
Absolutely. We're using this Stanford Encyclopedia philosophy entry on phylogenetic inference as our guide.
And it's fantastic.
It is, because it doesn't just give you all the biological details, Yeah. It actually highlights the deep philosophical debates that are happening beneath the surface of all this.
Yeah, and that's what we're here for. To
Exactly.
really unpack that stuff.
So get ready for some real aha moments, I think, where you're like, oh, so that's connected to these big philosophical ideas.
I love those moments. Okay, so let's just lay some groundwork here.
Yeah.
Phylogenetics, the study of evolutionary history, basically, and how all living things are related.
Right. From those tiny little microbes all the way up to the massive blue whales, how are they all connected?
And phylogenetic inference is like being a detective, trying to piece together that history.
That's a great way to put it.
And Darwin's theory of evolution really kicked this whole thing into high gear.
It did. It revolutionized the field.
Suddenly, scientists were trying to figure out, actual family trees, not just classifying things based on what they look like.
Yeah. And it's so interesting how Darwin's ideas kind of clicked with these existing ways of thinking about life, like taxonomy, for example, the science of classification, right?
Yeah.
And then there's this concept of homology. You might remember this. A homologue is let me see, the same organ in different animals under every variety of form and function. That's how Richard Owen put it back in the day.
Yeah.
And evolution, it gives us the why behind these similarities. Shared ancestry.
Right.
Which is totally different from analogy, right? Where things look similar because they've evolved similar solutions to similar problems independently.
Right, like a bird wing and an insect wing.
Exactly.
Both for flying.
Both for flying, but totally different evolutionary paths.
Totally. And this idea that life has this branching structure, this genealogical structure, well, that led to phylogenetic trees.
Yeah?
You've probably seen those diagrams.
Oh, yeah.
They look like branching trees, and they show how different groups are related to each other.
Family tree for all of life. Exactly. The branches, those lines, they represent ancestral lineages. And where they split or join at a node, well, that's a common ancestor, right?
Right.
That diverged into different groups. And knowing how to read these trees, that's important too.
Yeah.
We think of time usually moving left to right. But if you go right to left, you can see how recently two groups shared an ancestor. And that tells you how closely related they are.
Yeah, it's a cool way to think about it.
Now, this brings us to some key terms. These are really useful even from a philosophical perspective. First, we've got monophyletic groups, or clades. Imagine a complete branch on that tree.
Okay.
That's your clade.
Got it.
Includes an ancestor and every single descendant. All of them.
Okay.
This is important in biology because it represents a full evolutionary story.
Okay.
Then there's paraphyletic groups. Think of a branch where you've chopped off one of the smaller twigs. You've got the main part, the ancestor, and some descendants, but not all of them.
Right.
Like the old grouping of reptiles that excluded birds. Classic example.
That's a good one.
And then you've got polyphyletic groups. These are even more artificial, really.
Yeah.
Imagine taking some unrelated twigs from different branches and just calling it a group because, I don't know, they look kind of similar.
Right.
The explanatory power in biology, it really comes from focusing on those complete stories, those monophyletic groups, those clades.
Because if you know a group is monophyletic, it means that any shared characteristics, what we call homologies, were inherited from that single common ancestor.
Exactly. Like, think about fur producing milk, having 3 middle ear bones. Those are all uniquely found in mammals.
They are.
Tells us they're a clade. And these homologies, they form this nested pattern, just like the clades themselves.
Yeah.
Mammals are part of a bigger group called amniotes, which are part of tetrapods and so on, right?
Right.
It's this nested arrangement that led scientists to see life as having this hierarchical structure. Even Darwin himself, he saw this as a really strong piece of evidence for the idea of common descent.
Absolutely. Now, the way we go about actually reconstructing these phylogenetic trees, that's changed quite a bit over time, right?
It has.
For like a century after Darwin, it was mostly based on expert judgment, basically.
It was.
Someone with really deep knowledge of a group would just make a really educated guess about which traits were homologous,
Yeah.
how likely certain evolutionary changes were. And, I mean, there were definitely disagreements.
Absolutely.
Like, where exactly do the coelacanth and lungfish fit in with us four-legged land dwellers? That was a big puzzle for a long time.
Yeah, and what's important here is that while those early scientists, they did get a lot of it right,
They did.
But this reliance on individual expertise, it raised questions about how objective the process really was,
Yeah.
how rigorous it was. And this really came to a head in the mid-20th century with what we call the systematics wars.
Oh, the war.
Three main schools of thought really clashed in their approaches to all of this.
Yeah, so first we've got phoneticism, also known as numerical taxonomy. And these folks, they were a little skeptical, I think, about whether we could ever really know the details of common ancestry.
They were.
So they focused on just measuring the overall similarity between organisms based on lots of different traits.
Right. Measure as many traits as you can.
And group them based on who had the most similar sets of characteristics.
Exactly.
And they really wanted objective, repeatable methods
They get.
to keep those evolutionary theories separate from the active grouping.
Yeah, keep it objective. Then you had cladistics or phylogenetic systematics.
The cladists,
Thinkers like Willy Hennig. This was their big idea.
huge for them.
Recency of common ancestry.
Yeah.
That should be the only basis for deciding how related organisms are.
So stop.
They were really strict about it. All the groups we recognize above the species level, they had to be monophyletic.
Had to be.
This led to some pretty big revisions of traditional classifications.
It did. Like birds.
Birds, yeah.
They're not just related to dinosaurs.
Nope.
They are dinosaurs according to cladistics.
According to cladistics, they are dinosaurs. And if your definition of a dinosaur excludes birds, well, from a cladistic perspective, that makes dinosaur a paraphyletic group,
Right.
and therefore not a valid evolutionary unit for classification.
Interesting.
Then you had evolutionary systematics.
Middle ground.
Yeah, trying to find some compromise here.
Mm-hmm.
Thinkers like Simpson and Mayer, they wanted to incorporate evolutionary history into classifications, of course.
Yeah.
But they felt it was still okay to base those classifications on big adaptive differences,
Right.
even if it meant sometimes accepting some paraphyletic groups.
Right. So they might point to birds again.
Yeah, birds again.
Okay, they descended from reptiles, but they've evolved so much for flight
They have so specialized.
that it justifies giving them their own separate group, even if they share a more recent common ancestor with some reptiles than those reptiles do with each other.
That's the argument. And what's philosophically interesting here is how these schools, they were basically debating what even makes a biological classification meaningful at all.
Yeah.
Like, what are the fundamental principles behind a scientific classification system?
It's deep.
But in the end, cladistics, with its focus on monophyly, well, it's become the dominant approach in modern systematics.
It has.
It's powerful.
And it seems like the focus shifted from arguing about where to draw the lines in the classification system to actually figuring out the branching patterns of the tree itself.
Right. Like, once we have a good tree, who cares about the exact names and categories?
And Felsenstein had a great line about this. He talked about the "it doesn't matter very much" school,
Oh, yeah, he did.
where once we have a well-supported tree, the specific classification, it becomes less important.
Exactly. But, I mean, there are still debates about this.
There are.
Like those around the phylocode, for instance.
Yeah.
About how we should translate all this phylogenetic knowledge into an actual system of names.
But even within cladistics, there are divisions.
Yeah, and this is where pattern cladism comes in. Now, these folks, they see trees as summaries of character distribution, patterns of traits.
Yeah.
They're very cautious about making claims about evolutionary history or incorporating assumptions about evolutionary processes into their methods.
Yeah.
For them, a tree, it's more about relationships between the traits, not necessarily about, ancestry and descent.
And the split within cladistics between those who focus on process and those on pattern, I mean, it raises some big questions.
It does.
Ones that philosophers, I think, haven't explored enough.
You're right, like about the nature of scientific representation.
Exactly.
Inference in historical sciences, really
Yeah.
interesting stuff.
Yeah, why haven't philosophers jumped on this more? It's a gold mine.
I know, right?
Yeah.
It's all about scientific explanation.
Yeah.
The role of theoretical assumptions in shaping our methods.
Anyway, despite these internal debates, the systematics wars are mostly a thing of the past.
They are.
And the next big shift came with the molecular revolution.
Right. And I mean, if we just focus on the systematics wars, we might miss the huge impact of molecular data in computers in all of this.
Yeah.
The molecular revolution, it brought an explosion of data, especially for bacteria and archaea, where there aren't many fossils.
Yeah.
And it wasn't just the amount of data. Molecular sequences provided a completely independent source of evidence for common ancestry.
Right, you could compare it to the morphological data.
Exactly.
Right.
And at first, some of the more traditional thinkers were skeptical, but as more studies came out, the power of molecular data became clear.
It did. It could solve those long-standing puzzles, like relationships among turtles, and provide insights into those really, really deep evolutionary relationships.
Exactly.
And it's fascinating how molecular data, like DNA and protein sequences, they can be seen as more neutral.
Yeah.
They evolve more through random mutations and genetic drift than, being shaped by natural selection.
Right, so it can be less susceptible to that problem of analogy.
Yeah.
Those misleading similarities that arise from convergent evolution.
Right. And by comparing these molecular sequences, we can get a clearer picture of how things are actually connected.
Absolutely. And that's where the idea of the molecular clock comes in. Zucker-Kendall and Pauling, they noticed that homologous proteins in different species, they had these subtle but consistent differences.
Yeah.
And they thought, well, maybe the rate at which these changes accumulate is roughly constant.
Like a clock.
Like a clock, ticking away at an evolutionary pace.
So if you calibrate the clock using fossils, you can estimate when lineages diverged.
Exactly. And that even sparked debates about when humans and gorillas split, for instance. And this idea, it's even been applied to language evolution,
Wild.
It is. And the molecular revolution, it really opened up new frontiers, especially for those microbes.
Absolutely.
Carl Woese, his work with ribosomal RNA sequences, revealed that the archaea, they were a totally distinct domain of life.
They were.
Just as different from bacteria as we are. Completely reshaped how we view life.
And that wouldn't have been possible without molecular data.
It wouldn't have. And this is where the methodization of phylogenetics comes in, right?
Math.
Because molecular sequences are much easier to analyze mathematically compared to, physical traits.
Right.
It allowed scientists to build models of how DNA and protein sequences change over time, and then use those models to infer trees from huge data sets.
So we've got the basic toolkit now of modern phylogenetic inference.
We do.
We've got distance-based methods.
Like least squares, for example.
Yeah, which calculate the overall distance between sequences and build a tree that reflects those distances.
Yeah, pretty straightforward.
Then we have the parsimony-based methods.
Where you find the tree that requires the fewest evolutionary changes to explain the data.
Right, aligned with the cladistic idea of minimizing those ad hoc explanations for homoplasy.
Exactly. And then we've got the model-based methods.
More statistically complex.
They are. Likelihood and Bayesian approaches.
Right, so likelihood methods, they calculate the probability of seeing the data given a specific tree and a specific model of how those sequences evolved.
Yeah, and there are a bunch of different models, right?
There are.
From simple ones like Jukes-Cantor,
Yeah.
assumes equal rates of change,
Right.
to much more complex models like K80, HKY85, to Murray.
I feel like I'm back in bioinformatics class.
Me too, a little bit.
But a big challenge here is model selection.
It is.
How do you know which model is right for your data?
Right. That's a big philosophical question too, right?
It is. Okay, Bayesian methods, what do they bring to the table?
So Bayesian methods take it a step further. They use prior probabilities, or beliefs about the trees and the model parameters before we look at the data.
Okay.
Then, using Bayes' theorem, you combine that prior information with the likelihood of the data to calculate the posterior probability.
Right, the probability of the tree being correct given the data and your prior beliefs.
Right. And these methods really became practical in the 90s with algorithms like Markov-chain Monte Carlo or MCMC,
Right.
which allowed us to explore those huge numbers of possible trees.
Okay, so this background, it sets the stage for some really deep philosophical questions about phylogenetic inference.
It does. Because at its heart, it's about comparing different hypotheses, different trees, to explain the same data.
Yeah.
And that brings up the classic philosophical problem of underdetermination.
Right. Where multiple explanations can fit the evidence.
Exactly. So how do we choose the best tree?
Right.
And what's really fascinating is that figuring out what even counts as evidence, the characters we analyze, the way we code them, that's all connected to building the tree itself.
Yeah, okay, let's dig into that a bit. Identifying the evidence, what we call character analysis.
Yeah,
It can seem kind of boring, but some argue it's the most important part.
it is super important.
Character analysis is about building that data matrix, deciding what features are important, and then figuring out if a character in one organism is the same character in another.
That's the homology problem.
It is. And then you've got to decide on the character states. Like, if eye color is the character, then blue, brown, or green are the states.
Exactly. And philosophically, it's really interesting how we even figure out if a character in one species is homologous to a character in another one.
Right.
Early on, remember phoneticism?
Yeah.
They wanted to make character identification theory-free,
Right.
just based on similarity.
Mm-hmm.
But there are problems with that.
Yeah.
In phylogenetics, our understanding of homology is based on descent with modification. Similarity is evidence of shared history.
Right.
But these homologies also serve as the evidence for our phylogenetic hypotheses. So it's this like loop
It is a loop.
between theory and evidence, really important.
And this leads to those debates about how we should even define characters.
It does.
What counts as a single character?
Right. Like Richards, he argued that without clear rules, this process becomes subjective.
Yeah.
Others have tried to find more objective ways based on the underlying genetics of a trait,
Right.
or by just looking at what biologists actually do.
Yeah.
And then there's the difference between primary homologies.
Those initial guesses about what characters are the same.
Right, based on structure
Yeah.
and secondary homologies.
It's the similarities we infer after we've done the analysis.
Exactly. So it's this back and forth between observation and inference.
Okay, but molecular data, it's not always straightforward either, is it?
It's not.
With DNA, the coding is simple, right? A, T, C, G.
Right, 4 nucleotides.
But the problem is alignment.
It is. How do you know a position in one sequence corresponds to the same position in another one?
Yeah.
You can use conserved regions or functional similarity, but even those rely on assumptions about homology.
They do. And then
Yeah.
you've got gene duplication and loss, which complicate things further.
Absolutely. Make it really hard to trace the history of specific genes.
Okay, so even after you build your data matrix, there's another problem.
There is
The sheer number of possible trees.
so many trees.
As you add more species, it explodes.
Exponentially. It's a huge philosophical point, though.
It is, because finding the best tree, whether it's the most parsimonious one, the maximum likelihood one, and even aligning the sequences, it's computationally very challenging.
Super challenging.
These are what we call NP-complete problems.
They are. Meaning, as the data grows, the time it takes to find the best solution just blows up.
Yeah.
It becomes impossible to check every possibility, even with the best computers.
And Bayesian inference runs into this too.
It does. Even though it gives us that probabilistic framework, the computations can still be really demanding.
So researchers have had to get creative.
They have. They've had to develop these heuristic search strategies, basically clever shortcuts.
Right, to explore the possible trees without having to look at every single one.
Yeah, like in a Bayesian analysis, you might start with a tree that was made with a faster method, like parsimony.
To give the algorithm a good starting point.
Exactly. And this whole field of computational phylogenetics is a big deal in statistics and computer science.
It is.
And for philosophers interested in big data, it's a really cool case study of how scientists handle these huge computational problems.
Absolutely. So all of this brings us to the big epistemological question.
Yeah, what justifies our inferences? How do we know what we know?
And let's start with parsimony.
Okay.
One of the earliest methods, especially popular with the cladists.
Find the tree with the fewest evolutionary changes.
Right. And early on, the philosophical justification for parsimony was connected to Karl Popper's idea of falsification.
Right.
The argument was that each character is a hypothesis of homology, and the trees test these hypotheses.
Exactly. And the most parsimonious tree, it had the fewest homoplasies, meaning fewer falsified homology hypotheses.
Right. And how well that aligns with Popper is debatable.
It is debatable, but it was influential.
Very influential. And more recently, the focus has shifted to comparing parsimony and model-based methods in terms of falsificationism.
Yeah, which is more consistent with the idea of falsification.
Right. But parsimony has also been defended by appealing to, well, the principle of parsimony itself.
Occam's razor.
Yeah, the simplest explanation is usually the best.
And in phylogenetics, it means minimizing those ad hoc explanations for convergence or reversals.
Right. And there have been other philosophical arguments for parsimony?
Like Fitzhugh, he argues that phylogenetic inference is abductive.
It's about finding the best explanation for the patterns.
Right, not just statistical or inductive.
But Sober, he agrees that abduction is there, but he thinks it supports likelihood methods.
Yeah, that abduction itself justifies using likelihood.
Now, we can't forget the pattern cladists.
No, we can't.
They have strong objections to model-based methods.
They do, because these methods incorporate assumptions about evolution. They see them as flawed.
Right.
For them, parsimony is more objective because it just focuses on the patterns in the data.
Right.
They say you don't need to know how evolution works to figure out relationships.
And those who like statistical methods, they've argued that parsimony actually does rely on models, just implicitly.
They do. They say model-based methods are more transparent because they make those assumptions clear.
Right, and this is part of why the term cladist is so messy.
It is messy.
The editors of the journal Cladistics even said they're committed to parsimony.
Yeah, it highlights the philosophical divides in the field.
It does. So cladist, it's become a loaded term.
It has. Quinn has pointed out it can mean a lot of different things.
Yeah, like strict adherence to parsimony or specific philosophical views about classification.
It's a term philosophers need to be careful with.
Absolutely.
Yeah.
Okay, so now we have to talk about a big challenge to parsimony.
Okay.
Long branch attraction.
Ah, yes. This is a fascinating one.
Could you explain what it is and why it matters philosophically?
So imagine you have two lineages that are evolving really fast. Just by chance, they might end up with some of the same mutations.
Okay.
Now, parsimony, it wants the tree with the fewest changes, right?
Right.
So it might see those shared mutations as evidence that those lineages are closely related, even if they're not.
It's like the method is tricked by how fast they're evolving.
Exactly. And in these cases, Felsenstein showed that parsimony can be statistically inconsistent.
Meaning?
Meaning, as you get more data, it doesn't guarantee you'll get the right answer.
Okay.
In fact, with long branch attraction, parsimony can get more confident in a wrong tree as you add more data.
Wow. So it's not just inconsistent, it's misleading.
Exactly. And this was a big blow to the idea that parsimony was a really robust way to test hypotheses.
And later studies backed this up, right?
They did. In the 90s, they showed that parsimony and some distance-based methods were susceptible to this,
Yeah.
while likelihood methods did better.
And Felsenstein's work really emphasized statistical consistency as a way to judge methods.
It did. But not everyone agrees it's the most important thing.
Right, like Sober.
Yeah, Sober argues you can't dismiss a method just because it fails sometimes.
Right, especially in weird situations.
And he points out that likelihood isn't always consistent either.
True.
He thinks likelihood might be a more fundamental principle.
Like a basic assumption about how we should reason.
Exactly. And some who, like parsimony, have argued that the problem of inconsistency in real life is overblown.
Yeah, they say all methods can be wrong, especially if evolution is complex.
They do.
Wow.
And while likelihood is consistent if you have the right model.
Which we rarely know for sure.
Right, it's hard to know.
So phylogenetic inference, it's not just about algorithms.
That's not.
It's tied to deep questions in the philosophy of statistics and the philosophy of science in general.
Absolutely.
It's where biologists and philosophers are really wrestling with how we know things.
They are. And phylogenetic inference, even though it's rooted in biology, it has these connections to so many other areas of philosophy.
Yeah.
It's not just about knowing things about the past.
Right.
It's also about the nature of the things we're studying, what makes good science, the role of models.
For example, understanding evolutionary history, it's not just a biology thing, right?
No, it's not.
It connects to geology and paleontology, helps us date fossils, understand how life and Earth have co-evolved.
It does.
It shows how different fields depend on each other.
Yeah, it's a great example of that. And if we think back to the systematics wars, it's not just about classification.
It's about what makes good science in a historical context.
Exactly. And it wasn't always clear what the theories were behind each view.
Sometimes it was more about basic commitments to certain ways of thinking.
Yeah, different ways to evaluate evidence. And it's interesting how many researchers today use lots of different methods.
Looking for robust results.
Yeah, is that pluralism or is it just being cautious?
Interesting question. And then there's metaphysics.
Oh yeah, the deep stuff.
You might think it's totally separate from doing phylogenetic inference.
Right. Like whether species are natural kinds or individuals.
Uh-huh.
Does that matter for building trees?
But early phylogeneticists, they knew their metaphysical views mattered.
They did. If you think species are historical entities, that affects how you approach reconstructing history.
Right, compared to seeing them as just groups with shared similarities.
Exactly. And this still matters today.
Yeah, like the debate about birds and dinosaurs.
That's a great example. It's not just about relationships, it's about what a dinosaur even is.
Right, and Haber argues that the units of phylogeny, the things we're studying, they're best understood as units of divergence and diversification.
That's his view, yeah, connecting metaphysics to inference.
Really interesting. Okay, so now we have to acknowledge something.
What's that?
Those nice, clean phylogenetic trees we see,
Yeah,
they're simplified.
they are.
Evolution is often messier.
It is. There's hybridization, non-instantaneous speciation.
Right. Standard methods assume that the history of the characters perfectly matches the history of the species.
Right, one nice bifurcating tree.
But that's often not true.
No, it's not. Lineages merge, they swap genes.
Hybridization, integration.
Exactly.
Yeah.
And speciation can be a long process.
Not just a clean split.
Not at all.
Yeah.
And these complexities, like reticulation, where lineages merge, and discordance, where different genes have different histories.
They're becoming more and more obvious as we analyze more genomes.
Absolutely.
Ah,
And that has implications for how we think about phylogenetic inference.
it does. What does a tree even represent if things are so messy?
Right. Is it the history of the organism or just the average signal from all the genes?
And each gene might have its own story.
Exactly. And the concordance tree, that idea, tries to address this.
By focusing on relationships that show up across most of the genome.
Yeah. And in microbes, where horizontal gene transfers everywhere, it's even challenged the idea of a single tree of life.
Right. But these complexities also push us to develop better methods
They do.
by incorporating population genetics like coalescent theory.
Right, which helps us model the internal structure of lineages.
Yeah, and see how individual genes relate to the species' history.
Exactly. And Madison's work shows that discordance isn't just noise.
It can be real evidence of the evolutionary process.
Absolutely. And this shift from trying to force everything into a simple tree to dealing with the messiness, it's scientific progress.
It is.
We're building new models, new tools to deal with reticulation and discordance. Like the work on the tomato clade shows how this can reveal hidden parts of evolutionary history.
It does, but Sukumaran and Knowles, they warn us about inferring species just from these coalescent analyses.
Yeah, they remind us that it's about gene flow and reproductive isolation, not necessarily clear species boundaries.
Right, so we